Okay, we can definitely start. Thank you everyone for joining. I am Daniele Baldo. I am the communications officer for AgroServ, Horizon Europe project, which is lasting until 2027, and it's advancing the agroecological transition through integrated services with 12 research infrastructures that are uniting their forces to provide a comprehensive catalog of services and also connect research with practice through living labs and a really comprehensive effort all over Europe to advance the agroecological transition. This is a new episode, let's say, of the AgroServ Connect series, a series of webinars that we are organizing to showcase research that is happening within AgroServ, but also outside. And today we are joined by two users from the Transnational and Virtual Access Program of AgroServ. They are together today. They discuss similar topics in their research related to drought resilience, and the challenges that biotic and abiotic stresses represent for crops and harvests. They are online with us, Dr. Valentina Spanic from the Agricultural Institute Osijek, and Dr. Iman Mirmazloum from the Hungarian University of Agriculture and Life Sciences. They will explain what their research is about, and to different extents, they will also tell us how AgroServ impacts or can impact their research and their careers. So without further ado, I give the floor to Dr. Valentina Spanic, and afterwards the floor goes to Iman Mirmazloum, and after that we have a short discussion together, and we can of course take any question from the audience. So Valentina, the floor is yours.
Thank you, Daniel. Can you see my presentation?
Yeah, it works fine.
Okay. So first, just a few words from the institute that I'm coming from. So this is Agricultural Institute Osijek, and it's located in the eastern part of Croatia in a town called Osijek. So where actually the majority of wheat and maize is grown in Croatia. So we are also established in 1878, and we are doing breeding of different crops, such as wheat, barley, maize, alfalfa, pea, and soybean. So as I'm working on wheat, I'm coming from Department of Small Cereal Crops Breeding and Genetics. And at this department, we are running 3 different breeding programs for winter wheat, winter barley, and spring barley. Until now, we have more than 150 winter wheat varieties recognized to our institute. So beside these programs, we are also running different research projects, which are, for example, financed by Croatian Science Foundation or European Union or some bilateral projects and so on. And actually, we are working both on abiotic and biotic stresses and also on drought. But let me start now with our main topic, which is drought. And most of you know that climatic conditions today, they are actually producing different abiotic and biotic stresses. And drought is actually one of the most important abiotic stress today. So drought actually has a very high impact on grain yield and quality of wheat and other cereals. And this is especially important in the context of food security, because you know that the world population is growing. And this, of course, drought, as I told you, it has negative impact on grain yield. And also it can damage wheat plants at any point of growing season. But for example, specific stages such as seedlings, they are also very sensitive. For example, if we have drought in germination, then drought can reduce the germination percentage and rate. Then, for example, in wheat tillering, it can reduce the number of tillers. And then in flowering and grain filling stage, drought also can have very high impact. For example, it can interrupt grain filling, and that is why grain weight will be reduced, and of course at the end also grain yield will be reduced. For us, it's extremely important to find genetical drought tolerance, and because some studies they also confirmed that if some genotype has a tolerance in germination, it will also has a tolerance in later growth stages. Some previous investigations which we carried out, they also confirmed this thesis. We also we were very much interested about that how wheat cope with drought and how which system is working in wheat to manage the drought. And actually, it was demonstrated that antioxidant system and also some metabolites that they are occurring very much in response to drought. That is why we were interested in these systems, which I will show you now what we did in our previous investigations. So first, we carried out the experiment with drought simulation on wheat seedlings in plant growth chambers. So we established 3 treatments, where in each treatment we had 10 genotypes in 6 replicates. And first treatment was a treatment where we added polyethylene glycol in concentration of 10%. Second treatment was polyethylene glycol with 20% concentration. And third treatment were, of course, control plants, which were normally irrigated. After 7 days of simulation with drought, we measured different morphological traits, such as germination energy, percentage of germination, shoot and root length, dry and fresh weight of shoots and roots, and so on. And also we sampled the seedlings for further analysis with gas chromatography time of light mass spectrometry. Here are just some results. So in total, we analyzed 668 metabolites, and it was shown that 54 metabolites were significant between control and treatment with 10% polyethylene glycol, and 140 metabolites were significantly changed between control and treatment with 20% polyethylene glycol. After some further analysis, it was shown that there were the highest top 25 peaks in both drought treatments. And it was shown that oxalic acid, sulfurose, and turanose, they were increased in both treatments in compared to control, while some other metabolites such as butanoic acid, free uridiopropanic acid, tropic, glycine, and phosphoenolipuric acid, they were decreased. So if you are interested about these metabolites in seedlings, you can find this in our research articles which were published. But we didn't stop at this seedling stage, so we also did research in other growth stages, such as tillering and anthesis. And here I will show you the experiment for drought simulation with anthesis in controlled conditions. So we aimed here to quantify the effects of two drought intensities achieved by deprivation of watering for 45 and 65% of the volumetric soil moisture content for 14 days after anthesis. Especially here, at this stage, we wanted to identify physiobiochemical and molecular changes which are related to drought tolerance. And particularly here, I'm talking about antioxidant system and photosynthesis and some genes which are related to drought tolerance. Here I picked up some graphs, some results from this investigation, and I will show you just some. So here you can see two graphs where we measured catalase and glutathione reductase activity in 6 winter wheat genotypes. And here it was obvious that catalase and glutathione reductase, they were both significantly reduced in severe drought, and especially in drought sensitive and medium drought sensitive genotypes. So actually you can see on the graph, so first 3 genotypes, they were sensitive or medium sensitive to drought, and second 3 and other 3 genotypes, they were actually more tolerant or medium tolerant. Also here, I choose graphs with some genes which are related to drought tolerance. And here you can see the two genes which are related to dehydrins, such as DHN5 and VWZYV gene. And here you can see also that these 2 genes, they were especially increased in one tolerant, or better to say, the most tolerant genotype to drought. This is especially, it could be seen in severe drought. But now I just a little bit to conclude about our investigation in all growth stages. And we could see that extinguishing of chlorophyll may be stress indicator as first-line defense system in the flag leaves of wheat. And also we could see that malondialdehyde accumulation was increased. But on the other hand, we could see that, for example, carotenoids, they helped the plants to cope with drought. We also concluded that drought, of course, induced oxidative stress, and that beside catalase enzymes of ascorbat glutathione cycle, they also appeared to function as important components of antioxidative defense under severe drought. Here I also, as I mentioned at the beginning, and I am also stressing out now that drought tolerant and medium tolerant with genotypes, they could better acclimatize to drought, and they also induce antioxidative systems much earlier than drought sensitive genotypes. Also, I mentioned drought genes which are related to drought. They are also very important in tolerance of genotypes. Also, you can find these results in our articles which are published. Now I will transfer a little bit to our AgroServ project, which is titled Drought Stress in Winter Wheat, Unfolding the Challenges. First, I need to mention that so we have two service providers. So first provider is from Belgium, and this is a research institute for agriculture, fisheries and food by Belgium, which is belonging to Emphasis project. And the second one is from Italy, and this is International Agency for New Technologies, Energies and Sustainable Development, Biotechnologies and Agro-Industry Division, which is belonging to Metro Food project. And of course, users are we. And beside me also Dr. Jurica Duvnjak, which is postdoc, he will be also carrying out the research. So as I mentioned through my presentation, we could see that plants, they have different mechanisms in response to drought stress. As I mentioned, antioxidative metabolism, different metabolites, expression of different responsive genes, and so on, and photosynthesis also. And now we want to extend our research to metabolites in different growth stages. And that's why we will do our research. First stage will be done in Belgium. So where we will use automated rainout shelters, which will provoke drought stress. And this will last for 3 weeks in different growth stages of wheat. As I mentioned, the most important are sowing or germination, tillering and anthesis. And also we will have control plants where normal irrigation will be done. And in each growth stage, stress will last for 3 weeks. And we will measure different traits at the beginning of the stress and at the end of the stress. We will measure below ground and underground traits. For example, underground traits will be measured by drones, which will have different sensors. And below ground traits, they will be measured by AFT phenotyping. Also, now after each stress, we will do sampling of leaves and roots. And these samples, they will be sent to Italy, where actually metabolomics research will be done. And we agreed that we will measure around 100 semi-polar and 60 non-polar compounds. And this study, for sure, of these genetically diverse genotypes, which will be under drought in different growth stages, will provide new, very important insights into plant responses, which will also include different biochemical pathways that are responsible for adaptation to drought stress. As I mentioned, we will use different genotypes, and we will see which genotypes is the most tolerant, and we could see which mechanism is responsible for this tolerance. And actually, this will help us in our breeding program to find more and more tolerant genotypes. And actually, overall, this should have impact, also socioeconomic impact, but also we expect scientific impact, as we will also, we plan to write articles where also other researchers could see what we will do and what will our results be in this AgroServ project. And actually also this is a multidisciplinary research, which is also very important, and also 3 different institutions, they are connected somehow, and maybe in some future we will do some other researches, but overall this will help us all, we hope. And at the end, of course, I want to thank you for your attention.
Thank you, Valentina. Thanks a lot. Very, very interesting. Also to see how you connected your previous publications and research to the access you're obtaining through the 2 research infrastructures from AgroServ. So I urge the audience, if you have any question, you can use the Q&A section here in the chat. If you want to start thinking about any kind of question. I also forgot to mention that here with us today is Eba Ibrahim, which is our central access manager in AgroServ. She's also going to be available afterwards if you have any question on the access on the currently open transnational virtual access call that we have in AgroServ. But now, Iman Mirmazloum, the floor is, of course, yours. You can go ahead with.
Thank you for this invitation. I hope you can hear me and see the slide.
Yeah, we see the slides. Yes.
Okay, great. And thank you, Valentina, for this interesting talks. So you just saved me some time about talking about the drought stress and the importance of that. I am working in a Hungarian University of Agriculture and Life Sciences. We are located in Budapest, and we have several campuses in different places in the country, covering multidisciplinary research related to the life sciences, ranging from agronomy, horticulture to food science, and even landscape architecture. So that's where I work, actually. And today I'm really thankful to Daniel and the AgroServ team that they allowed me to present some slides from what I was thinking about the biostimulants and the research that I proposed to the AgroServ and the conducted activities so far in the past, I would say 9-10 months. Talking about the drought stress, I don't want to repeat too much of things, but we are all bombarded by this alarming and this yellow, orange, and this kind of maps all the time, that what's going on in the world related to the climate change and so on. Just to briefly put some numbers here. So the drought stress, as Valentina mentioned, is one of the most serious abiotic stresses that are affecting mostly the agricultural sections. And if we look at the distribution of the maps that they're presenting, usually the countries that are in this belt here at this central belt, so the countries that are mostly producing the foods, cereals and so on, and they are actually the subject mostly to the predicted severe drought in the future. And if we look at the publications that are addressing the droughts, we can see really a dramatic shift and an increase, really significant. So, and if you look at the other charts in the down corner, so most of them are coming from the Asian and South Asian countries. So the high populated areas of the world, and they really need to do something about it because the food demand is really in stack. I found this bibliographic study related to the drought stress, and interestingly, so if you can see these categories, that I think these are the base data based on the Scopus website, and so the legit scientific publications are considered. And you can see these yellow parts here down, if it's visible, and these are the ones associated to the strategies. So of course we know that the research on drought stress is going on for a long time. And mostly it was focused on the physiology, abiotic stresses, genetics, as you can see, photosynthesis, as Valentina also mentioned, an important factor. But the strategies to put together these findings, which are really piling significantly, is scarce. Back to my activities. So when we were doing research on drought stress in 2010, I think, so that was quite a kind of treatment and not really a kind of concern. So we were doing research to see that if we can give drought stress to plants to somehow increase the contents of the added value components. Let's say in this case of silibomarium, for example, we were looking to increase the polyunsaturated fatty acids. And if you look at the trends in the publications and research, what we published last year or this year in 2025 in this article, so we are not really considering those type of research anymore, but we are really concerning about the lack of food and how an integrative approach is needed to really address the drought stress, because now the food is in demand and increasing or decreasing some active metabolites may be still important from the research point of view, but not a global concern, I would say. So there was really a significant shift in the area of research related to the drought stress, I would say. And this research, this is a paper with like 76 contributors or co-authors from 14 different countries, and we all agreed that one single line approach is not really sufficient or efficient in the era that we are living, but we need a really multidisciplinary approach, an integrative approach, and hopefully by using the AI, because the data that are emerging in this line of research are really expanding very rapidly, and we have a lot of data that are produced from different perspectives, so from breedings or speedy breedings or different areas. But what we need is really a kind of prediction modeling, or to do some kind of modeling to predict the occurrence of the drought stress, and that's where the AI can come and then somehow help us to do the management to finally to reach to the climate-proof crops that is aiming nowadays the research. Back to the biostimulants, which was the topic of my AgroServ project, I just thought that first I need to do some introductory update about the current landscape related to this term. Actually, we know that the companies, it was interesting for me, I have to mention this, I was checking the websites of Biostimulant World Congress, which will be, I think, in November, December in Spain. And the list of the participants was really surprising for me. Enormous amount of companies are present in that conference or seminar, I would say. And I guess all of them would present some biostimulant products that have been developed or worked on for several years, I would say now. But a bit of the history, that how this became really such a trendy topic nowadays. I would mention that the first time that the biostimulant name appeared was in the early 90s. And the definition, which was formed in that time, was based on some non-fertilizer products that have enhancing effects on the plant growth if they are grown under suboptimum conditions. So under, let's say, the abiotic stress or mostly abiotic stresses, so that was a definition that was given to this kind of products. And if they are applied at low concentrations and in advance, so prior to the stress. So with that one, which was really, I think, based on one single paper, which was published in 2015. And if you can see here, the number of citations, more than 3,300 times this paper was cited. And that's where really the, how to say, unified definition of the biostimulants was agreed on and founded. So, and so on from that time on. So we stayed with this concept of non-fertilizers, but function enhancers as biostimulant. And this concept was actually substantiated the current regulatory networks of the European Union, which are stating that these products or biostimulants are the ones that are stimulating plant nutrition processes independently of the product's composition or content. So what is inside really doesn't matter according to the regulations. Of course, it matters if they are direct nutrient or plant growth regulators, hormones, but mostly the function is important. And this is a so-called claim-based model, which is a bit actually different than the American definition of the biostimulants, which also, I mean, they differ slightly in the case that they are also considering the growth enhancement as there is also biostimulant. But according to the European ones, so these are the products that would help the plant to better survive the utility or the utilization of the nutrients and to cope with the stresses and also to help with the bioavailability of the nutrients in the rhizosphere. I encourage everyone in this forum to look at this editorial article, which was really insightful. So this Unlocking the Black Box of Plant Biostimulants, I think this is a continuation of that 2015 article, which is a bit updated and really shows the concern about the research on the biostimulants era. These are some key points that I wanted to define here. Excuse me if this is full of text, so there will be only 4 of it, and after that we will shift to some colorful slides. But these are really important from the point of view that the definition of the biostimulants really matter. So we are still living in the area that also the confusions which is coming from the terminology is something that can be the source of misuse by any sections or sectors in the agriculture, and also slows down the research and adaptation. Because when we are considering, and I will show you some slides to clear this concept, that what do I mean by the confusion in the definition, and that is very important to consider. Another aspect which we need really to cover is the concept of beyond yield. So it seems that, or it's, at least we are, to our understanding, we are trying to reflect that the biostimulants are not really there to increase the yield, as it was advertised in several places, but they are really, looks like priming agents that would somehow secure the plants or prepare the plants to get to the stress. So when the onset of the stress is coming, the ones which are treated with the biostimulants are supposed to perform better comparing to the non-treated one. And this means that we are not really looking in the immediate increase in the biomass, but we are building a kind of long-term resilience or resistance. And also important to note that, so they are not just a new inputs like different types of fertilizers that we were applying so far, but they are a kind of shift to enhance the biological potential of the plant. So they are a kind of hint that if the plants receive them, then they take it as a message or a signal to really trigger some physiological processes inside of their tissues or cells, then they would be more ready to deal with the stress which is coming. And so this one is a kind of shift, and also the transparency. So we really need to clear and define what biostimulants are actually and what they are not, and how they really work. What is really or mostly missing from the products is the mode of action or the mechanism of action. Of course, some can argument that according to the European regulation, that's not really important. If there is a trial in the field and a product was showing some improvement in the stress tolerance, then that's enough. But to my view, I think the producers are, even if they don't tell it, but inside, they really need to know that what is inside of this product that would give me this outcome, if they give, and then what is really the mechanism of action behind it. So these are the ones that we really need to consider. Back to this article which I mentioned, I just borrowed this table from that. So this is a categorization of the mode of actions of the biostimulants. I'm not going to read it through, but I just put it here. So the main categories are the nutrient use efficiency, abiotic stress tolerance, crop quality traits improvement, and the nutrient availability in the soil. So if we consider only these criteria, then we have some innovation challenges. For example, in the regulatory documents, they're not mentioning of the new biopolymers or nanoparticles that are really emerging from the research and slowly are entering the market, because we still don't know that what is the role or what is the, how to say, the regulation about using this green synthesized nanoparticles that are used for, let's say, a better delivery or slow release of the active compounds that are present in this product. And then there are some emerging technologies, so you still don't know what would be the regulations about, for example, the nanocarriers for double-stranded RNA deliveries. So again, this is a new era that we know that for the biotic stress protection, so that has been tested, so we can just send some RNA molecules to interfere with the expression of the genes that are associated to the defense mechanisms, and those appears under nature. So again, we have some blurry areas in this. Also the same for the nanocellulose, nanoketones as a carriers or coating agents, which is turning out that they also have some biological activities themselves. So somehow the borders are a bit not clear, that needs to be really defined. So my point is here that the research is really going in a speed that the regulations are somehow behind, and that there should be a kind of balance in this. And this is what everybody who is interested in field, I think, should consider it. These are some examples about the biostimulants, for example, these mycorrhizal fungi, microbes, or humic acids. And so I just categorized this. So I put some examples here for each of these claims, which was in that table in the earlier slide. And then there are some examples of them, that how they were blamed that they can help the plant to cope with the abiotic stresses. Some mentioning about the novel biostimulant classes that are emerging in the research, at least, and they're showing some potential. These are, I listed a few of them. Of course, the list is not limited to these, but at least these are the ones which I found based on solid research in the literature. So, for example, ectoins or polyamines or polyestrols, for example, or nanocarrier-based biostimulants. So these are, as I mentioned, targeting the delivery of RNA molecules or some kind of endophytic consortia or marine-derived compounds. So these are, again, some trendy biostimulants that are gaining some claims. But again, mentioning that regulatory issues which I mentioned, if we just consider, let's say, these phytoestrols, and there are literatures that they are helping the plants to tolerate the drought stress. So I just put 3 of them here. But if you consider, for example, the campestrol, which is a precursor of the plant hormones racinosteroids, again, according to the regulations, a product containing the plant growth regulations cannot be marketed as biostimulant. So again, there are this, if there is a precursor of a compound, is it really considered as a hormone, or what would be the regulations if there is a product containing a precursor of a plant hormone? The other 2 are not known to be involved in that BR biosynthetic pathways, so there is still potential for them to be freely used. Even some more novel biostimulants I encountered, so these are emerging now, and I found them really interesting for the future research. I just mentioned, again, a few of them for those who were thinking about the future research. So these are fungal cluster filtrates, so plant cell culture extracts, these nanofiber nanoprimings or nanoformulations for a precise delivery of the active compounds or active peptide enzymes. Actually, these are the ones that would definitely shift the boundaries of the definition of the biostimulant. So these are enzyme-based inputs that would help the plant to enhance photosynthesis or root growth, and also some biological nitrification inhibitors that would protect the soil nitrogen by supporting the activity of denitrifying or nitrifying microbe. So the list is going on. With this, I try to summarize that what is the landscape of the research. If you can see the publications on the Scopus with the keyword biostimulants, it really jumped significantly, and we can see that there is a shift, let's say, from humic acid now to the microalgae. So the yellow ones are the dominant publications based on the Scopus, and are showing that there was really sustainability or protein hydrolysate. So these are the ones that are gaining more attention, and of course associated to climate change. I skipped this one. But this one, so the biostimulant, it's a kind of categorization. So the visualization of the co-occurrence of the keywords in more than 4,000 publications that I could found in 2010 to 2025. And then we can see that really there is a link between the abiotic ester. So the bigger the circle is showing that the occurrence of the keywords in those publications were more and more significant. So biostimulants and abiotic esterases are definitely the ones that is in the focus of the research. But to show something that really a biostimulant or a compound or any kind of treatments on abiotic estrase tolerance is really meaningful, I thought that I'd take one example of our research. It's not about the drought, actually, totally the opposite, the flooding stress on cucumbers. And I'm just showing a few slides about this because I didn't want to have this presentation really pure science, but just to give some visual view that what we are talking about an evidence-based trial. So what do we really mean and what is the expectations of the growers? This one here, the A is the control plants, so cucumbers like 1 month old, and here in the middle we have a flooded one, so you can see really a difference between the treatments and the controls. And then we have here nitrate supply. Nitrate is a fertilizer, so again we will see more that what would be the problem with the definition of the biostimulants. So the nitrate was given to the flooded plants, and then here we have a scavenger of nitrate derivatives, one of the final products of the nitrate after nitrification, or nitrate, nitrite, and nitric oxide, one of these derivatives. And we can see that if we really remove or scavenge the nitric oxide, we are really removing the positive effects of nitrate in this system. And I'm showing this to show that how we can really go and see what's going on in the system. So we have the tools nowadays, so the research infrastructures are ready to measure several metabolomics and even at the gene expression level. An example here, we can see, okay, we can confirm that the hydrogen peroxide level was increased after the flooding, and then it was decreased after the nitrate, and again, a slightly increased when the effect of nitric oxide was gone. The same is for the malondialdehyde and let's say a marker of the lipid peroxidation in the plants, usually coming from the oxidative esterases, and in this case we are losing the effect again. If you look at the nitrate, so we can really trace it if the nitrate which was given to the plants have been really transported to the tissues, and then we confirmed that yes, and it was really gone there up to the leaf, to the roots, and also the nitrate reductase activity enzymes were. So all of them were correlating that there is something going on with the nitric oxide, and finally the nitric oxide content in the root was really boosted because of the nitrate. So here we are showing that really the nitrate here is not acting as a fertilizer, but it's really acting as a donor of a signaling molecules, which is nitric oxide. And if you look at the gene expression data, I didn't bring the whole set of the data, but the paper is available, so the link is here. If somebody is interested, you can look at it. But then we will see the nitrate transporter genes that are mostly upregulated when we are giving the nitrate, and then when the nitric oxide is missing, again we are losing that higher expression. Some of the genes that are responsible in the nitric oxide metabolism, let's say this RAP2, for example, is coding for a acetylene response factor, again involved in the hypoxia reaction of the plants, and these RBOH genes also they are involved in the production of reactive oxygen species, this respiratory burst oxidase homologs, and so on. So from the morphology to some physiology and then to some molecular levels, so we are able now to really pinpoint that what's going on when we are giving a compound to the plants. But when it comes to the definition of them, so can I consider again this nitrate as a biostimulant? According to the regulations, no. But then we know that some of the, let's say, biostimulants that are in the market containing some, let's say, seaweed extracts or microalgae extracts, and these extracts are also full in nitrate. So maybe there is the nitrate within these extracts or biostimulants that would act again to produce this nitric oxide and then to trigger these whole signaling pathways that are there. Again, I didn't want to escape this one because this is an interesting, again, a study. So we have two different types of cucumber here. What you are seeing here is a nightshade system, which is reflecting the biophoton emissions of the plants, and usually associated, or we believe that it's associated to the lipid peroxidation. And these signals are coming after some dark adaptations. just to compare them, so these are two different hybrids of cucumbers. One of them is the open field and the other one is the greenhouse type. And here we try to give them higher concentration of nutrition. Let's say the whole-gland solution was the case here. And we can see clear differences between the two cultivars to react to the higher nutrition solutions. So that's, again, supports the idea that why some biostimulants are working for some crops, but not for the other one. Although it's now we are showing that within the species we have different reactions. I think I have to skip a few slides here, and this one is showing that the abiotic stresses are not really an isolated event. So this is an interesting paper showing these different letters, that how many central metabolites in the plants are affected in common when there are different abiotic stresses: cold, drought, flooding, heat, and salt. So it's really an interconnected thing. So I'm not concerned at why I showed you a flooding example instead of a drought, because mostly when we are talking about the stresses, we are talking about some complex systems that are mostly in common in different types of abiotic stresses. Back to my AgroServ TA call, and which was accepted like last year. I think I was one of the first applicants to this, and I'm really grateful for that, which was giving me a really huge opportunity. We were not even thinking to do such a large scale experiments in different countries, but I'm just showing here the map that how many institutes were involved in my proposal, and I was really lucky to meet brilliant people during these visits. So I will show you a few, a journey-like report that it was started in Greece, in Heraklion, this Marine Research Institute of HCMR, and here you can see the Manolis Mandalakis, who was in the contact person that I visited there. So the idea here was to find some microalgae or seaweeds that are not, cultivated, or somehow they are considered as waste materials, not a waste, but somehow easily available as a cheap source of biostimulants. And we agreed on 2 species, and we did some extractions with different methods, and then we somehow could manage to get the extracts of these 2 species that I would use them in my next steps. I will be a bit faster. I'm not going to show you that why we use, let's say, this species, this Positonia, but these are the wash-ups, let's say, in the shores of Mediterranean seas. There are arguments that we shouldn't remove them because of the ecosystems and so on, protecting the shores, but still, I think a lot of them will be dumped and, let's say, converted to compost or different purposes. And then we thought that maybe we can get some extracts with some biostimulants activity in it. And it turned out that, yes, there are some potential in it. And then my next visit was in Luke, in Joensuu, Finland, when I met Dr. Timo Domic, a brilliant researcher. So we also tested some of these biostimulants along with other ones which I had. And then we tested the cold stress, let's say, in their beautiful root lab in UN. So nice experience. I will be a bit fast because the time is running out. And then my next visit was in Belgium, in Louvain-la-Neuve. So where I learned really how to deal with the mycorrhizal fungi. These are vascular mycorrhizal fungi. When I received also a strain from them, we had several meetings and discussions to choose one of the strains of their huge collections with the potential for plants which I was planning to use, and I received them. So the research also started in that line. I will show you a few photos from what we did in the lab. So the mass production of the spores here, you can see in the second one that this is a non-inoculated maize plants, and the yellowish one is the inoculated one. And then we tried to use it in our basil plants to see if we can really detect or see the associations of these mycorrhizal fungi with the roots of our plants. And we did some microscopy, and it turned out that yes, the system is working. At least now we have an established system. We know how to deal with them. So it's, I don't know if it's visible or not, but here you can see, let's say, they are vascular, exactly similar to what we are seeing in the illustration or scientific illustrations. So I hope these are a bit inspiring, and then having these different aspects of biostimulants and some preliminary studies on phytotoxicity effects of these compounds and so on. Finally, we did these main experiments, or the biggest one, I would say, in Vienna Biocenter. Here you can see the basil plants and the cucumbers that are grown, and some photos I just showed here for inspiration, that we had really a good time, great time, talking about the challenges, solving the problems, unpredictable problems that would emerge. And finally, here in the lower one, this is the sampling days of our plants in Vienna Biocenter. I just put these slides here. I have a few more. I try to be on time, but the blue ones are just showing that, for example, if I want to use this chitosan nanomelatonin SMP as a biostimulant, how many types of control do I have to include in my, let's say, experimental design? So that's what a proper experiment needs to look like, I would say. Of course, we're still missing one compound here, the antagonism of the melatonin receptors, which we couldn't get on time from the companies. But so I'm just showing the extent of that. If you want to show really the effect of one biostimulant, you really need to consider this type of, or this much of controls to show really that this formulation has an effect. I also had the chance to get some biostimulants as biomass of some algae production. So this is a collection in Hungary from Dr. Molnár Zoltán. I encourage anyone who is interested in the microalgae and cyanobacteria to contact him, because they have a huge collection of more than, I think, 200 or no, more than 1,000 live cultures of this. A few images. Now we are left with, like, I think 10 terabytes of data, as they told me, and now we are starting to analyze really the effect of more than 30 biostimulants that we tested on cucumbers under flooding conditions and on basils under drought conditions. So hopefully we will be able to analyze this data and come up with the final conclusions. I want to close this with this slide, which we know that only 30 to 50% of the irrigation water actually reaches the plant roots. So this is a generalized quantity in the world. And we know that roughly 30 to 40% of the food product is lost and waste before they use. And I asked the ChatGPT a question, and this was the results, that in a well-designed plant experiments with low variability and adequate replications, a treatment-induced increase of around 10% in agronomic or physiological traits can often lead to statistically significant differences. So this is what you can publish. So if you see really a 10% increase in the physiological parameters, you can publish it and you can claim that, yes, this was effective bias. But we shouldn't really forget that if we want to address the food shortage globally, we need really to focus, again, in a multidisciplinary approach, integrative approach, improving the irrigation systems and improving the food distribution system and so on. And I see only the future that the AI or this artificial intelligence are coming in to help us to be more efficient in this era. I will close it with introducing our a special issue in a journal of plant stress. So still the submission is still open until the 30th of September. If somebody has some research related to this, I encourage you to look at this. And finally, I thank you for your attention and listening. And I'm sorry I'm like 6-7 minutes due.
It's fine.
Thank you. Thank you.
Thank you, Iman. Thank you very much for the presentation. Very comprehensive on your research and your access that you've been carrying out through AgroServ. Very, very interesting to see how you put together, in your case, 4 different research infrastructures with different facilities and services that you implemented through your application is very, very interesting. I would ask Heba Ibrahim, our Dr. Ibrahim, our Central Access Manager, if she has any comment on the 2 presentations. I see she already left a question to Valentina. I don't know if Valentina, you're still with us. she has a question on the
Yeah.
I don't know if you saw the question on the challenges. What kind of challenges you think you are going to face in setting up your experiments that you have to do?
Yes, thank you very much for your question. I can see now. So as I mentioned in presentation, so this is extension already of our research. So till now we did metabolism in seedlings of wheat, and now we will extend this to other growth stages. And also till now we did not use any rain shelters. So all our researchers were in controlled conditions or in greenhouses or in plant chambers, or we just did field experiments. So this will be novelty to do it in rain shelters. And as I mentioned, also metabolism will be carried out in Italy at different growth stages.
Okay, so AgroServ will push you to new boundaries, let's say.
Yes, hopefully.
Okay.
And we are particularly interested because we are working on varieties which were produced by us, and that's why we are also interested in this kind of research and to find drought tolerance.
Okay.
Thank you very much, Valentina. It's really very nice to see the impact of our services and of AgroServ on your research, and we look forward to see the output and the outcomes of your research.
Yes, we are also looking forward. And also we are looking forward to collaboration with Belgium, institution in Belgium and also in Italy.
I also have a question or comments for Dr. Iman as well, if I may take the floor again. So I'm really also very impressed, and I liked your presentation. I find it very great to see such a large project making use of several of our services in your research. I think you have 4 or 5 services in this project, right? Very good. I'm also curious to know, beyond the scientific results or publication, do you see any already potential of your findings to be transferred into practice? For example, supporting farmers or informing agricultural policy, so on.
Yeah, definitely. I mean, this is a valid question, and I think we are doing research finally and ultimately to come up with the products that would come to the market. And hopefully, yes, as you mentioned, publications are in the picture, but beyond the publications, so we are seeking really cooperations with the companies who are developing the biostimulants. Sometimes they're coming to us as a research institute, and they're asking if there are some feasibility in the products that they are thinking. And with really having this, how to say, from our own research, the data that are showing the potential, then we can really shape the formulations that they are thinking. Sometimes, because I escaped or I excluded the, let's say, this mycorrhizal fungi from my experiment in Vienna, because we discussed that this is not a feasible way in this short term. So that was also a kind of, how to say, learning, that if a company is coming with a solution, then we have to look at it multidisciplinary from different aspects, from how is it feasible, really, the, let's say, the shelf life of their product, what kind of compounds are in it, and if we can pinpoint a mechanism which is associated to one of these active ingredients in this kind of formulations, and then we can suggest them that, yes, that's the one which has feasibility, and then they can really go on to the production and trials and the fields. But I have to really mention that this, I forgot to mention, when we are talking about the fields, so we are talking about multiplex environmental factors that would influence the outcomes of such a product. And this one, we cannot really neglect it unless we have some predicting models developed based on a global data, I would say, to somehow summarize it. Yes, this biostimulant product in this context, in this stage of the growth would work, or possibly would work. Yeah.
Very nice. Thank you very much. And I just wonder if Valentina or Iman, any of you will be at the AgroServ conference in October. No.
I won't, unfortunately.
No.
Perhaps another event. Maybe
yes,
we can have a more elaborate
or
discussion.
remotely, if it's possible. I would be pleased to. Yeah.
Yes, me too. Hopefully some other time.
Absolutely, we will work to before the end of the project. We work to have a more comprehensive event for I would say all users, but probably that's going to be difficult. But everyone involved in AgroServ, it would be very nice to have a collective exchange of experiences and results. So our time has run out, unfortunately. I would just like to remind you that the AgroServ Access program is currently offering an open call. The deadline is set for October 15th. So if you have any question, you can probably find your answers on the AgroServ website related to how to access and what are the eligibility criteria. You can find also contacts directly to the central access manager, so HEBA, directly on the website. If you're interested in applying, you should do so very soon. You should take contact with the facility managers of the services that are offered within the catalog of services. You have my email here for any kind of extra question. I can relay you to the different facilities that are within AgroServ. So again, thanks both to Valentina and Iman for taking the time. Thank you, Eba, for being with us. We didn't have a lot of time to assess any kind of question from the audience, but anyway, we are here. You can reach out to me, Heba, and the whole AgroServ team. So I wish all of you a good end of the day, and see you very soon.
Thank you, Daniel.
Thank you, Daniela. Thank you, Iman and Valentina.
Thank you very much to all of you.
Thank you, everyone.
Bye, everyone.
Bye.